All right. Exam on Friday. Any questions on any of that? Okay. Well, I'll be looking at my email more frequently into the evening. So if, as you're studying, something comes up that you need some clarification on, go ahead and send me an email and I will try to get back to you as soon as I can. Um, and let me know also if you want to meet up for office hours. Okay. I can see what we can come up with if my office hours don't work. Okay, so this stuff today, of course, is not going to be on the exam. We're getting into the second part of photosynthesis, where the plant is taking in the carbon dioxide and making sugars off of it. So that's the Calvin cycle. We've worked through that now, all the way from start to finish. And so this pie chart idea breaks it down into three major phases: carboxylation, reduction, regeneration. And if you think about the first step where you see the CO2 coming in through the stomata, then you get this reaction going on where it reacts with that 5-carbon sugar, RUBP, that has 2 phosphates on it. And you have that really unstable molecule that you don't need to know the name of, but then you end up with 2 identical molecules of phosphoglyceric acid, or PGA. And in order to get carbon dioxide and RUBP to react with each other, you have to use the Rubisco enzyme for that step. So remember, Rubisco enzyme, the CO on that acronym, stands for carboxylase oxygenase. So it's saying it's a dual functioning enzyme. ASE is an enzyme, a word like that is enzymes. And so we have, right, our Rubisco is capable of bringing in CO2, and so that's the carboxylation. It can bring in CO2 to its active site, but it can also bring in oxygen. It's not very picky. Either or will work for Rubisco. And then that has severe consequences for what that reaction will be and what the products will end up being. And we'll get into that in a little bit. So that's the first stage. And we noted no energy. You don't need ATP. You don't need NADPH. If Rubisco sees enough carbon dioxide coming at it and enough RUBP, it's just going to go ahead and react those two. So that's all good. And you end up with that lovely PGA. But then the next step does need an input of molecules that are coming from the light reactions. Remember, this is all happening in the stroma of the chloroplast, that watery portion. And that's exactly where NADPH and ATP are formed. So that's convenient. And so with the reduction, you have to NADPH will reduce. So that's why they call it the reduction phase. They're kind of intimating that NADPH is involved. But don't forget about ATP, because you need to have another phosphate group as well in this reaction. So this is the reaction where you have, they're only showing one, but you have 2 of these PGA molecules. And you see that actually first it gets phosphorylated and then a reduction step. And so that converts PGA to G3P, glyceraldehyde 3-phosphate. So you get 2 molecules of those. And so that's the reduction phase. So step 2, that reduction phase, yes, you need the light reaction products in order to get that to go to completion. So then once you get that G3P, then that's kind of like a branch point of what's happening. You get some of that G3P sent out to the cytoplasm. And that's what they're showing here. If it gets sent out, it goes through a rearrangement to DHAP, and then it gets sent out into the cytoplasm, and you get the formation of, there's a bunch of reactions that will make the sucrose. Or it could stay if this is really cooking along really well and there's plenty of G3P building up, then the plant might go, you know what, I think I'm gonna make some starch. So they're also showing that. So that's another option. So if everything's going great, the plant will say, yeah, I'm gonna go ahead and save up for a rainy day. I'm gonna make some starch. And that just stays in the chloroplast. It'll keep it in the chloroplast. You'll actually see some starch granules in the chloroplast if you look at it under the microscope. So that's kind of a branch point idea where you have some options of what's going to happen to that pool of G3P that's building up over time. But most of it actually has to go into the third phase of the Calvin cycle or C3 cycle, which is the regeneration, because you have to have enough RUBP all day long in order for this to happen, you know, to keep it going over and over and over. And so as long as the stomata are open, CO2 is coming in, the plant's going to make a really concerted effort to make sure there's plenty of RUBP. So that regeneration phase is the biggest portion of this pie chart. And we get quite a bit of stuff going on here. I like to teach off of this one. So here, it's the whole cycle. Here's the carboxylation event right here. Whoops, with PGA being formed. And then the reduction is right there with PGA getting converted to G3P. But then all the rest of this is regeneration, essentially. Whoops. Okay, so most of what you're seeing is the regeneration. So you have, here's that branch point idea where maybe the plant's going to send some G3P off. So when it says export, that means it's going into the cytoplasm. And that will be for sucrose synthesis. Then here you're seeing the flow where the plant may say, you know, I'm going to make some starch, save up for a rainy day. And so I'll just keep it in the chloroplast, but I'm going to make it into starch. And then the rest of the G3P being shown on here is entering into the regeneration. Actually, we're going to show how these two G3Ps are going to react to the fructose and then keep going into regeneration. So it could go into starch or it could go into regeneration. So basically, the game is get to 5 carbons, because that's what RUBP is. So you want to get to 5 carbons. And so all of these reactions that you see are accomplishing that goal eventually, but it takes a little bit. And so we went through last time how there's that numbers game that I'd like you to kind of zoom in on, and it is vastly simplified. I don't need you to know all the enzymes or the intermediate product names, except for a couple of them, okay? So if you define that the 3 carbons is a G3P, you're using that G3P pool. If you combine 2 G3Ps, you're up to 6 carbons. And then if you take another G3P and attach it, you're up to 9 carbons. And then that molecule will quickly break down into a 5 and a 4 carbon. So 5 carbons, good. 4, we're not quite there. Somehow we got to get a 5 carbon out of that. So what happens is you get another G3P reacting with that 4 carbon. You're up to 7. Another G3P comes along. Now you're up to 10. And those break down into 2 5 carbons. So that's good. So now we have those 3 5 carbons, but they're not quite there yet because there's only 1 phosphate attached to those 5 carbon molecules. So you got to put in some ATP in order to fully form the ribulose bisphosphate. Bis means two phosphates. You got to tack on some more phosphates to each of those 5 carbons. Okay, so this is basically what I'm looking for if I ask about the regeneration phase. Okay, you don't need to know all the enzymes or intermediates. So 5 is the magic number, and once you get that happening, then here you can see that you end up with, here's your, whoops, let me do this. xylulose 5-phosphate, ribose, xylulose. Okay, so all of those are rearranging into ribulose 5-phosphate. So that's what I mean by rearranging. And then you can see the ATP reacting to tack on another phosphate. Okay, so the Calvin cycle, if everything's going okay, you have to imagine this cycle zinging around, so to speak, all day. Yeah, let me go back into this. Okay, go back to this one. Okay. So if this cycle keeps zinging around and around and around all day long, you can see how eventually you're going to get a decent pool of G3P that can then get siphoned off, so to speak, into making sucrose, which of course is hugely important for the plant because that's the form of the carbohydrate that is going to send through its body to nourish other parts that can't do photosynthesis well enough or can't do it at all. Okay, and then don't forget, I did include some other PowerPoints that if you like to do carbon counting, or if this works better for your brain, they're in there. That's fine. So if a plant is running the Calvin cycle, and this is their way of making sugars, you can imagine that's really important and that they have to have some way to tightly control it. I'm not going to go and explain all the ways that they control it. I'm going to give you a couple of examples. But basically, there's this process that they've discovered called autocatalysis, where at night, when the sun goes down, the plants aren't going to do photosynthesis at night, not the plants, especially around here. So they're going to close down their stomata, kind of go to sleep, so to speak. Well, then you can imagine, especially RUBP is going to start to go down, levels of it, the concentration of it is going to start to go down. So then you can imagine, well, okay, then when the sun comes up, there could be too little RUBP for the plant to be really efficient, at least at sunrise. And so basically what they've noted as they look at how the plant is doing this through the night, they actually will divert and make sure that they have enough RUBP built up. And so they will run some special physiology at night in order to make sure that when the sun strikes the plant the next morning, they have enough RUBP, like a big pool of it ready to go. And so they do have that mechanism happening during the night just to make sure that they're all good. So that's one example. The other idea is that when the sun goes down at night, and especially in total darkness, you won't have certainly the light reactions going on. You won't have that because there's no sunlight. And you also don't have the making of sugar going on at night. That all just fades away. And the reason for that is that several of the enzymes that I didn't ask you to know about, except for Rubisco, several of the enzymes in the Calvin cycle, they're actually what they call light dependent. They need light in order to fully function. And so I want to explain a little bit about that. So far, they know there's 5 enzymes that are activated this way. And one of them happens to be Rubisco. So I'll talk about Rubisco kind of separate from the other 4. So if you look at the other 4, let's do those first. What you see on their enzyme area is that they have this sulfur disulfide bond that will get cleaved through this special system called the ferredoxin thioreducin system. So ferredoxin, that should ring a bell. That is a player in the light reactions. Okay. So this ferredoxin thioreducin, what this is just a transfer of electrons. It's a redox reaction going on. Okay. It's a little short one. And basically the end result is that you get a break of the disulfide bond. And once that happens, then you have the ability of that enzyme to fully function. So basically what this is saying is because ferredoxin is involved and ferredoxin is part of the light reactions, that's how ferredoxin gains an electron is through this electron transport process. And in order for ferredoxin to get an electron, you have to have the light reactions going on. You have to have light doing its thing to excite the electrons and send them on down the electron transport chain. Ferredoxin gets it. Then in this case, instead of ferredoxin giving it to FNR, you will have it do something like this. So here they're trying to show this idea that it's light activated. So this right here is the artist's abbreviation of the light reactions. Okay, and then they say, okay, now ferredoxin gets the electron, so ferredoxin reduced, that's through the light reactions. It grabs an electron through the light reactions. And then what you have to do is look at the arrow coming off of ferredoxin here. How it swoops like this. What they're trying to show is that ferredoxin gives it to ferredoxin thioreduxin reductase. And when that gets the electron, it breaks that disulfide bond. And so now you have those sulfurs reduced. Okay. So there's this swooping, so to speak, of the electron, and that will reduce those sulfurs. And then it goes on, little short transfer of electrons until you see finally you get the fully activated form of the enzyme. So basically, they're saying that it's light activated because you have to have ferredoxin kicking this off, and ferredoxin doesn't get an electron unless you have the light reactions going. So that's their logic of that. So at least 4 enzymes in the Calvin cycle work this way, where they will be quiet in the dark, but they'll kick into full gear through this little pathway, this redox system. This is another look at it where they're trying to show, the dark inactive, the light, then it's active. And then here's another glimpse at it from a different source, saying the same thing, trying to intimate that, yes, you definitely need light energy for ferredoxin to get reduced. Okay, so 4 enzymes at least can do that process. Rubisco is a little different. It's also activated by light, and this is through a different system. So we know that this is the enzyme that kicks off the very first step of the Calvin cycle. So its job is to take CO2 into its active site and react it with RUBP. What they've also discovered is that there's a second carbon dioxide that they've named the activator carbon dioxide, and it binds to a lysine portion of the active site. So this is not the carbon dioxide that reacts with RUBP. This is a separate one. And once this second CO2 comes in and binds to that lysine, then they refer to Rubisco as the carbamate form. Okay, so that second CO2 comes in, that activator CO2 comes in. Then the next thing that happens is magnesium comes in and binds to that activator. Then Rubisco is fully active. So if you don't have this happening, Rubisco is not going to do its job. So how can this be light activated? This is a little more challenging concept here. So basically, I've tried to draw it here. Okay, so up top is the Rubisco on the far left. They're saying this Rubisco is in its inactive form. There's the lysine that's, supposed to be in the active site. And here here's your activator carbon coming in, and it binds to the amino group of the lysine residue, the lysine amino acid. So that's the carbamylation reaction, okay, in the carbamate form, but it's not quite done. It has to then get a magnesium attached to that activator carbon. Okay, so how is this light activated? All right, so this is happening in the stroma of the chloroplast. Here I'm trying to, this green oval is the thylakoid membrane floating around in the chloroplast. Light energy strikes it, you get the kickoff of the electron transport chain. One of the other things that's happening during the electron transport chain process is proton pumping, where you have protons at key points during that electron transport chain. Protons get pumped from the stroma to the lumen. Well, okay, so then you get a bunch of protons building up. What that does, which I haven't really talked about to date, but what they notice is as the protons are coming in, that allows for magnesium to come out of the lumen. And then the magnesium attaches to the carbamate to form, and now Rubisco is fully functional. So they're saying that without light and proton pumping, you don't get magnesium coming out and attaching to Rubisco in the stroma. So it's kind of this domino falling impact where you have to kind of walk back a few steps to see this process, okay? So light energy is necessary for that proton gradient to get established. The proton gradient is necessary for the magnesium to move into the stroma from the lumen. And then it can make the Rubisco fully active. Okay. So light activation. Then one other thing I'll just mention, Rubisco also is regulated by this enzyme called Rubisco activase. So what happens at night when it's all dark? Rubisco gets phosphate groups attached to itself. And well, they're sugar phosphates. I like to think of it as like a big fluffy blanket of phosphate, sugar phosphates that are covering Rubisco. And it's nice and cozy. Rubisco just goes to bed. And that is the signal for Rubisco. Okay, chill out. It's dark. We're done. And so those sugar phosphates kind of blanket the Rubisco, stop it from being active. And basically, what's really happening is it stops from that carbon dioxide coming in and the magnesium coming in. Then, right before sunrise, they notice Rubisco activase getting active. And it's named Rubisco activase because it does activate. What it does is it clips off all those sugar phosphates. Kind of reminds me of when I was a teenager and my dad would come into the room and turn on the light and take my blanket off if I was being really resistant to getting up for school. Time to get up. All right. So taking off those sugar phosphates, and then, of course, with sunlight striking the plant, it's good to go. It's going to get up and running. You'll get the carbamylation and the magnesium and all of that going. And then Rubisco will be fully active. So it makes sense that Rubisco, it has more than one way to kind of regulate it and make sure that it's acting when it should act and going quiet when it should go quiet. And same with, they're still trying to work through all the enzymes in the pathway. They suspect that most of the enzymes are going to be controlled pretty tightly because this is such a pivotal, important physiological process for the plant. Okay, so Rubisco. Rubisco, yeah, it's a different enzyme. It can cause this situation. Of course, they have to give it big, long names. C2 oxidative photosynthetic carbon cycle. Or they also call it the photosynthetic carbon oxidation cycle, which abbreviates down to PCO, or photorespiration. So all of those mean the same thing. I'm going to go with the short one and call it PCO because I don't like saying all that other stuff. So we're going to say the PCO cycle. I need to talk to you a little bit about this because Rubisco is kind of an odd duck when it comes to enzymes because, like I said before, it doesn't seem to care what comes into its active site. If carbon dioxide comes in, yeah, great. I'll react that with RUBP. If oxygen comes in, yeah, I'm fine with that. And that will get connected to RUBP. And so it's this dual enzyme. It's never evolved the ability to have a strict or tight binding affinity just with CO2. Like a lot of enzymes have really high binding affinity. Like I'm only going to take these two molecules in my active site and that's it. Here it's taking three things in. It's taking RUBP, or it could take carbon dioxide, or it could take oxygen. So what they have found out is it is a competitive reaction. What that means is basically if Rubisco is surrounded by lots of carbon dioxide, that's what comes into the active site. So carbon dioxide wins and it gets reacted with RUBP, great. That's the C3 cycle kicking off. If Rubisco, on the other hand, sees a ton of oxygen around it, and then that's what comes in to the active site. And it will react with RUBP, and then we don't have the kickoff of the Calvin cycle. So that's a problem. You can imagine where the plant's like, ooh, okay, not quite what I'm going for here. So if Rubisco has a bunch of oxygen around it, then the oxygenation reaction occurs. And this is what we get. We get RUBP and oxygen reacting. You do get one PGA, which is great. That can feed right into the Calvin cycle. That's part of the Calvin cycle. But then you get this funky phosphoglycolate. This is a two-carbon molecule because obviously oxygen doesn't have a carbon. So we're short a carbon. So you get one PGA and then this funky two-carbon phosphoglycolate. There are no enzymes in the Calvin cycle that can deal with a two-carbon. So this two-carbon is a problem. That cannot feed right into the Calvin cycle. Instead, we're going to see it feeds into this other really involved cycle. And the plant can end up managing to salvage and make a PGA out of this, but you'll see it's not easy. So but remember that PGA that also gets made, that can go right into the C3 cycle, and that's great for the plant. So then that two-carbon phosphoglycolate, that feeds into the so-called PCO cycle. Now, when you look at the entire cycle, the PCO cycle from start to finish, you can say that, well, they put it this way. They say it allows for partial recovery of carbon. What that means is you do end up making a PGA from that process, which is good. But some of the carbon is so-called lost along the way. During that cycle, I'll point that out. And then there's extra energy that the plant has to use in order to get this to go to completion. So when they say that some carbon is lost, what they actually see is that carbon dioxide will get formed instead of fixed. So carbon dioxide will get made in this cycle, and then it seems to just, because it's not in the chloroplast where this is happening, it gets respired out by the plant. Just like you're respiring CO2 right now. Okay, so it doesn't get fixed into carbohydrate, I should say. So because of that, they call it photorespiration, because you're doing respiration, you're giving off CO2. Well, this is the plant's version of it. And so this is not ideal. You don't want the PCO cycle running in a plant all the time. It's extra energy. You get the loss of carbon instead of getting it fixed into carbohydrate, it's just respired. So when they do experiments in the lab and they're running, they force a plant to do the PCO cycle, like long term, they see its growth rate slows down, its overall biomass at maturity is much smaller. If you're growing crops or something edible, the yield of, the edible portions is much lower. So in general, it's not a good thing to have running continually or frequently. So here is a mock-up of kind of the either or option, where you have RUBP sitting right in the middle here, and it's saying if it takes CO2 and reacts with CO2, so you have to imagine Rubisco sitting right here. So if you have that, that's great because you get the kickoff of the Calvin cycle. And of course, through this whole process, the plant is giving off oxygen, during photosynthesis. Here, on the other hand, they're showing what happens if oxygen comes in and reacts with Rubisco in the active site, and then RUBP and oxygen react. Then you do get one PGA, but then you get that phosphoglycolate. And then you actually get a loss of CO2. It's respired out. So they're trying to show this either or idea. So here, just if you want to count carbons and convince yourself, this is the reaction of RUBP and oxygen. And so instead of getting 2 molecules of PGA, you only get 1, and then you get this funky 2 carbon. And again, there's no enzyme in the Calvin cycle that can deal with that and convert it to PGA. So this gets shuttled over into the PCO cycle. And the PCO cycle is not straightforward. Here, this is just another look at the either or option. So they're showing RUBP, and they're saying if CO2 comes in, you follow what's happening here, and you get those 2 phosphoglycerate PGAs. If oxygen comes in, then you end up with 1 PGA and then that 2 carbon. So whatever Rubisco sees more of, that's what's going to come into the active site. If oxygen comes in to the active site, then the PCO cycle happens. This is the PCO cycle. I like to teach off of this one because it's simplified. This is also the PCO cycle, but you can see that it's a lot more detail if you put them side by side. But it is nice if you want to count carbons and convince yourself of what's happening here. So, I mean, it's a nice diagram, but I find it a little busy for teaching purposes. So I'm going to go back to the more simplified one here. So there's a couple of key things on here to think about. It's complicated, first of all, because it's not all happening in the chloroplast. You have 2 other organelles involved. So let's start at the chloroplast though. You have oxygen coming in, and so you have to imagine Rubisco sitting right here. And so oxygen comes into the active site, RUBP comes into the active site, and you get 1 of the PGAs formed. So they're showing, yep, you do get a PGA, and that can immediately feed into the C3 cycle. So that's good. So that's one of the products, but the other product is that phosphoglycolate, which is only 2 carbons. So you see now it gets banished out of the chloroplast. You got to go out, you got to find the peroxisome. Okay, so it will go through a short reaction first and form glycolate. So that phosphate gets cleaved off. Glycolate remains. Glycolate has to find the peroxisome. And whoops, and you see a series of reactions here. But basically what you see is the glycolate gets converted to glycine. And then glycine has to find the mitochondria. And now, once it gets in, there's a couple of things going on here. So the way the artist drew this, if you look at the mitochondria, you see that glycine I just circled. Then if you look over here, there's another glycine. What they're trying to say in this way of depicting it is that you actually need two glycines coming in to continue on. So basically what that's saying is the PCO has to run twice. You have to get two glycines coming in to the mitochondria in order to do the next series of reactions. I think this more complicated diagram is showing two of everything, two glycines coming in. Okay, so it's the same idea. The artist depicts it a little differently, that's all. So basically, we have two glycines have to make it into the mitochondria, and then you see these reactions going on where a couple of major things, well, one huge thing going on, really, there's your CO2 being given off. So CO2 gets made in the mitochondria. Instead of CO2 getting fixed into carbohydrate, which is what the plant's trying to do, it's actually being given off. And it's over in the mitochondria, that's just respired. It's a loss. That's what they call the carbon loss. And then you see that when you cleave off a carbon dioxide with these two glycines coming in, you end up with a serine. Well, now serine's got to go find the peroxisome, go through a series of reactions, turn into glycerate. Well, that's close. Glycerate's close to phosphoglycerate. So we're getting there. But now glycerate's got to get out of the peroxisome, go and find the chloroplast. And then if you phosphorylate it, which is being shown here, so that there's your energy, your extra energy, then you get back up to a PGA. So this is the partial recovery that they're talking about. It's partial because you did lose a carbon dioxide out of the process. So it's partial recovery. Yes, you do end up making a PGA out of this whole big thing. But there's a cost, ATP cost, and then the CO2 being given off instead of getting fixed into sugars. So complicated in the sense of lots of intermediates here, lots of stuff going on, 3 different organelles. So this also takes time and just physically to get everything where it needs to go. So you can imagine if you force a plant experimentally to do this for good stretches of time, this is a burden for the plant. So in general, plant physiologists say this is not a good thing for plants to run continually. Now, there's still a lot of debate because, you got to wonder, why didn't Rubisco ever evolve to be better, have a tighter binding affinity? It's weird. And so there's been a lot of debate about that because Rubisco's been around for over 2 billion years. So that's a lot of time, and yet it still is sloppy. And so that has led some of the plant physiologists to say, well, I wonder if maybe parts of the PCO cycle, maybe it does need to get run every once in a while for some plants. Maybe some of these intermediates are important. Maybe there's something more here that we're not seeing. We don't understand it. So they're still debating this and looking into it more. But the upshot is, yes, Rubisco is sloppy because it can take either carbon dioxide or oxygen into its active site. So we talk about the PCO cycle having to run twice because you need those two glycines coming into the mitochondria. So we say for every 2 turns of the PCO, you end up with 4 carbons from that if you look at the glycines, but one of them gets lost in the form of CO2, and then you get 1 PGA that can get sent into the C3 cycle. So these 4 carbons one of those carbons is given off as CO2, and then the other 3 carbons will make the phosphoglycerate. And the PGA, that can go right into the C3 cycle. So as far as thinking about when you're studying the PCO cycle, you don't have to know step by step, but I would like you to know the 3 organelles involved and know which molecules are like mobile, I guess, which ones are moving. So for example, it's the glycolate that comes out of the chloroplast and gets to the peroxisome, which could, and then the glycine gets made, which then has to find the mitochondria. And then 2 glycines get, CO2 comes off of that, and you end up with a serine that has to find the peroxisome. Finally, you get glycerate that has to find the chloroplast. And then knowing that you have ATP involved, energy cost, knowing that you get loss of carbon, CO2 given off. Those are huge. And then, of course, once PGA is made, that's good because then you can get the Calvin cycle cooking along. Okay. Any questions on the PCO cycle? Okay. So from the plant's perspective, when would the PCO cycle start to kick in? And a couple of things to think about here. First, understanding that when you have an aqueous solution like the stroma in the chloroplast, okay? And if you think about basically room temp, 25 degrees Celsius, and you look at the ratio of how much CO2 to O2 is in that aqueous solution, it's about 3 to 1. So the stroma, the aqueous portion of the chloroplast, has a 3 to 1 ratio, more CO2, 3 to 1 ratio. So that's good. That means that under these conditions, there'll be more CO2 than oxygen. And then that should favor the Calvin cycle. So, and again, if there's some situation where oxygen goes down, that's good because then you'll get CO2 winning the competition and the CO2 comes in to the active site. So you can think of it that way. If CO2 levels are high or if oxygen is low, that's all good. If oxygen levels go up a little bit, then oxygenation is favored due to how CO2 and oxygen interact with water. Oxygen is a little better here in terms of how it interacts with water, so you'll get that favoring. Also, if you have an aqueous solution where you have CO2 and O2 dissolved in the stroma there, but you start to increase the temperature of the chloroplast, the plant, essentially. If they get hotter temps around them, what you need to know is that the solubility, SOL, the solubility of gases decrease, which just means those gases are going to want to bubble out of the stroma. Okay. However, because of the chemistry of it, oxygen doesn't bubble out as fast. CO2 bubbles out faster. And then that's lost to the plant. Okay. So that means oxygen sticks around longer during hot temps. So if a plant is experiencing hotter temps, it's losing the ability, CO2 goes away, essentially. Oxygen is still there. So now we have the PCO cycle kicking in. Okay, so if you are a plant and you think about their day-to-day life, like around here in this area of the world, we get pretty decent temps, hot temps in July, in August. And most of the plants around here in July and August, for stretches of the day, they're dealing with the PCO cycle. They have that unfortunate situation where their body temp has gone up high enough where Rubisco is seeing more oxygen instead of CO2. And then the PCO cycle starts running. Now, if you go to really hot places, like a tropical forest near the equatorial line, where it's can be hot every day, you would think they're probably running the PCO cycle all the time, every day. But it turns out they're not. Because they managed to evolve a situation where they've solved this problem. And so we'll talk about that as we get a little bit further into this. But the other thing to think about, when you think about when Rubisco evolved a couple billion years ago, it came about in an atmosphere that had plenty of CO2 and no oxygen initially, or very, very little. So it never really had to worry about oxygen when it first was evolving and becoming active in photosynthesis. So that's something they often point to. Some scientists also are saying, you know, maybe some of those intermediates of the PCO cycle are important during certain times or phases for the plant. So we should look at that more carefully, because Rubisco on all these years of evolution it just hasn't evolved to be really tight for CO2. Some people say it's a safety valve for excess ATP. That one I don't really buy, but some people say that somehow that's protecting the plant by getting rid of it. I don't see that myself. I'm not convinced, but that's what some people say. So anyways, there is this idea that Rubisco's sloppy. It's never really learned or evolved to only take in CO2. And it takes in both. And so there's still quite a bit of debate about that. There's a couple of other enzymes also that are like this, but Rubisco is, you know, the most abundant enzyme on the face of the earth. And so it's really kind of weird. Okay, so what we need to think about now is like the plants along equatorial lines where they have hot temps pretty much every day. And yet when they study those plants, they're not running the PCO cycle, or at least not very much. And so there's another mechanism in play here that allows those plants to adapt to those parts of the world that are pretty routinely hot. So we'll talk about that next time. We'll get into that, but that won't be till Monday. So Friday, I'm going to try to get here at 10 to the hour in case this room is emptied out quick, and I'll try to get us in here as quick as possible. And then I can stay a little bit beyond 1 if necessary, because there's no one in here after us. Okay, so that's the plan. So come prepared to ask questions. Remember to ask questions during the exam. Nothing is supposed to be tricky. So just if it's confusing to you, make sure to ask questions.